Neil Clark, and this is part 3 of the series of talks discussing the characteristic direction, which is a geometrical approach to differential gene expression. In the last few lectures we've described what we mean by the characteristic direction and showed how we calculate characteristic direction. And now we want to find a way of showing that the characteristic direction is a good way of examining differential gene expression. Now the way we're going to do that is we're going to compare it to a selection of commonly used gene expression, differential expression analyses. The first is the oldest, the fold change, which is very simple. It just looks at the ratios of gene expression between two states. It takes no account of structure in the data or anything like that. The second is to use the student t-test, or the Welsh t-test. This looks gene by gene, and it makes the assumption of a normal distribution and provides a test for the difference of location of the gene expression values. And the last two are significance analysis of microarrays and LEMA, two very popular methods for analysis of differential gene expression. Now, the main problem we have in evaluating these methods of differential gene expression is that we don't have a gold standard. We don't know what the right answer is. We don't know which are the differentially expressed genes, usually. So one possible approach is to not use real data, but use synthetic data, which has the advantage that we know exactly what the differentially expressed genes are because we tell them to be differentially expressed, but it has the very strong disadvantage that the synthetic data only has the structure that we give it, and it lacks the rich structure of real biological data. So what we've done is we've devised a way which is a compromise. We give up on the idea of a gold standard and replace it with a silver standard. We've identified a situation where, although we don't know what the differentially expressed genes are, we do have some clue about what they are. And this is in the setting of transcription factor perturbations. So what we've done is we've compiled a large number of experiments where we have gene expression profiles from a control state and from a state in which a single transcription factor has been perturbed, for example by knockout. Now the reason that we have some clue about which differentially expressed genes, which genes are differentially expressed in this situation, is that we also, for each transcription factor, have ChIP-seq data, which tells us the DNA binding regions of the transcription factor. And if we can go and look closely at these DNA binding regions, we can associate some of these regions with genes. For example, if a binding site is in the promoter of a gene, we might consider this gene to be more likely than most to be differentially expressed upon perturbation of that transcription factor. So the first thing we do is to evaluate a method of differential gene expression, is we rank the genes according to the method's perception of its significance in the differential gene expression. So here in this movie, we can see that we've moved the most significant gene to the top of the list and the least significant gene to the bottom. Then we use the ChIP-seq data to identify genes associated with binding sites of the perturbed transcription factor, and we examine their position in this ranked list. Here we've indicated them with arrows and highlighted them as red. Now the hypothesis is that if a method is doing a very good job of analyzing the differential gene expression, these genes, highlighted red, should tend to be towards the top of the list, because these are genes which should be more likely than most to be differentially expressed upon perturbation of the transcription factor. Another way to look at it is to think, if we give up on doing a good job of the differential gene expression and we just randomly sort the genes, then the genes highlighted red would be sort of uniformly scattered throughout the list, and so there would be no tendency for them to be towards the top. So then what we do is we take this list and we lie it down flat, and we're going to walk from left to right along the list, and we're going to keep track of the cumulative distribution of the recovered transcription factor binding site associated genes. So the idea being that this is going to quantify the degree to which the differential expression analysis has ranked our red genes towards the top of the list. So you can see in this figure, the dotted line is what you would expect if you randomly sorted the genes, and the solid curve is the curve that you'd expect if your differential expression approach has preferentially prioritized the transcription binding site associated genes. Then what we can do is we can simply subtract off the random part to really sort of highlight the non-random part. And then what we're going to do is we're going to take this peaked distribution and we're going to associate the peak with a quantification of the quality of the method of differential expression analysis, because this measures the degree to which the method has prioritized our genes highlighted in red there. So here we can look at some results from some real data taken from GEO over many experiments perturbing transcription factors. We've used two different libraries of ChIP-seq data, one from CHIA and one from ENCODE studies. Both show similar results. So what we can see here are each line corresponds to a different approach to differential gene expression. If we look at the green line from the t-test, doesn't seem to be finding a signal, but the purple line from the t-test seems to be prioritizing genes in the differential expression which are associated with binding sites of the perturbed transcription factor. This occurs to a slightly greater degree with the SAM method and the Lima method, but to a much greater degree with the characteristic direction. So this provides us some evidence that the characteristic direction might be a particularly good approach to differential gene expression. So out of interest, we tried this also, in addition to transcription factor perturbations, we tried this for drug perturbations. And so we took gene expression data from control samples and from samples perturbed with a drug, and we looked for the differential expression of the known targets of that drug. And this is illustrated on the left here. You can see none of the methods find a really clear signal in this case. However, if we go to the protein-protein interaction network and find protein interactors with the known targets, then we get a much clearer signal. And the different methods have a similar ordering in terms of this measure. All these validation methods were performed using microarray technologies. There is currently a lot of effort to try and find the best statistical approaches in the newest RNA-seq technologies, and this is what we're going to discuss in the final part of this series.